Welcome to the Deep Dive, your fast track to becoming fluent in the world's most complex research. Today, we are tackling, well, a really fundamental truth about cancer. It's never static. We often try to classify tumors into these neat little boxes, clusters of cells. But the reality, it's much more dynamic, a fluid thing, what researchers call a phenotypic continuum.
Yeah, that adaptability is, it's the core challenge we face in oncology, really. Cancer cells aren't just, type A or type B. They're constantly shifting their function, adapting to survive stress or low oxygen, maybe nutrient shortages. So defining that whole spectrum of what they can become and doing it with computers, that's been, well, almost impossible.
Right, and if you can't even map that spectrum, how can you possibly target its weaknesses? Which brings us actually to the tool we're deep diving into today, AAnet. That's Archetypal Analysis Network. We're looking at a paper that introduces this machine learning tool. It's specifically designed to solve that computational puzzle defining cell states in this continuum, using triple-negative breast cancer, TNBC, as the main example.
Exactly. And the goal here is pretty revolutionary. Instead of just seeing cancer cells as these fixed clusters, the idea is to see them as a continuous blend of extreme specialized states. We call these extremes the archetypes, or ATs.
Okay.
Think of it like this, maybe. Imagine the entire universe of possibilities for a cancer cell. From like maximum growth speed to maximum survival mode, the ATs, they're the fixed specialized corners of that universe. And every single cell in the tumor is just some mixture, a percentage, technically a convex combination of those specialized corners.
Okay, let's unpack the computational headache first then. Why do our standard tools fail? Things like clustering that gives us cell type A, cell type B, or trajectory inference, which tries to find a nice straight developmental path. Why do they kind of fall apart when you feed them single-cell data from a really complex tumor?
Well, it really boils down to geometry, believe it or not.
Yeah.
Those standard techniques, they need clear boundaries between groups, or they assume a fairly simple linear path from one state to another. But single-cell data from cancer, it's incredibly complex, highly nonlinear, and frankly, super noisy.
All right.
Imagine trying to map the surface of, a crumpled, staticky towel. Standard methods just fail because they basically assume the towel is flat. When the data manifold, that's the sort of underlying shape of the data, is that complicated, they just can't figure out the rules of the cellular state space.
Okay, so that's where archetypal analysis, or AA, comes in. It's a type of factor analysis, right?
Yeah.
Designed to find those extremes you mentioned.
That's right.
But traditional AA has this big mathematical catch, doesn't it?
It does, yeah. Traditional AA is great at extracting the archetypes, the ATs, and it describes every cell as that blend of ATs, which biologically makes a lot of sense. A cell sitting right near the proliferative AT corner is highly specialized for growth. A cell far from any corner is maybe more uncommitted, a generalist. But here's the catch you mentioned. Standard AA assumes the data has to fit neatly inside a specific geometric shape called a simplex, right there in the original high-dimensional data space.
A simplex. Okay, that simple shape like a triangle or maybe a tetrahedron in 3D. Why is assuming the data looks like a neat triangle such a big problem for real, messy cancer data?
Because real biological data is almost never that simple. It's curved, it's folded, it's complex. So if your data set looks more like, say, a banana or a helix, and you try to force fit a flat triangle or a simple tetrahedron around it, well, the corners of your simplex, your supposed archetypes, might land in empty space where there's no actual data. Or worse, they might misrepresent the true extremes, maybe picking up some random outliers instead.
Okay. Traditional AI kind of struggles when that shape assumption doesn't hold.
Exactly. It can end up identifying meaningless points as these super specialized states.
So this is where AAnet, the neural network part, comes in. It's the fix. If the data is crumpled, AAnet has to somehow uncrumple it. How does this autoencoder thing work its magic?
Yeah, that's a good way to think about it. AAnet is essentially an autoencoder, which is a type of neural network good at learning representations. But instead of forcing the raw, complicated data to fit the simplex shape directly, the AAnet encoder parts learns the best way, the optimal nonlinear transformation, to sort of warp that complicated high-dimensional space until the data fits perfectly and neatly into a much lower dimensional, simplicial, latent space. It learns how to uncrumple the towel, basically.
Okay, that sounds like a really clever fix for the geometry problem. But hang on, if AAnet is effectively warping or manipulating the data to fit its geometric needs, how do we know it hasn't, distorted the picture? How do we trust this new map it creates?
That's a really important question. And that's where a key robustness feature comes in. AAnet doesn't just start from random guesses for where those corners, those archetypes might be. It uses something called diffusion extrema to get started.
Diffusion extrema.
Yeah. These are extreme points that are intelligently inferred from the underlying geometry of the data itself. They kind of capture the global structure, the main features of the true data manifold, even with all the noise.
And why does using those matter? Does it stop the network from, I don't know, finding fake extremes or getting lost in the noise?
Precisely. Using diffusion extrema gives the network really solid initial estimates for the ATs. It makes sure the network optimizes towards real biological extremes, rather than getting stuck chasing noisy artifacts or maybe just a few random outlier cells, which, as you know, is crucial for single-cell data where noise can be a huge problem.
Okay, that makes sense.
So the final output you get from AAnet is this thing called archetypal affinity, basically.
Yeah.
Scores for each cell telling you exactly how strongly it's committed to each of those extreme states, each AT.
All right, so we've built this pretty sophisticated mapping tool. Now for the big question. When you point this thing at TNBC triple-negative breast cancer, which is known for being aggressive and really varied, what actual corners pop out? What are the specialized states? The researchers used single-cell RNA sequencing data from metastatic TNBC cells, looking at the primary tumor and where it spread. So what 5 main specialized states, or ATs, did they find in those primary tumors?
Yeah, so AAnet identified 5 really distinct archetypes in the primary tumors. And what's cool is that each one lined up nicely with known functional cancer hallmarks. We can even give them colors to help remember. First, there's the proliferative AT, blue. This is basically the growth engine.
Yep.
It's often the most common AT they found, really driven by fast cell cycle progression, I think G2M checkpoint stuff, high CDK1 expression. Its main job is just divide, divide, divide.
Got it. Blue is growth.
Second, the oxidative adipogenic AT, yellow. This one's geared up for high energy production using oxidative phosphorylation or PHOS. It's also linked to stress responses like ROS production and, oddly, adipogenesis fat cell development pathways. This hints it might be vulnerable to drugs targeting fatty acid metabolism.
Yellow. High energy, maybe fat metabolism.
Third, the hypoxic AT, orange. This is the survival specialist, found in areas with low oxygen.
Ah, the tough conditions.
Exactly. So to cope, these cells ramp up oxygen-independent glycolysis. You see more enzymes like HK2 and LDHA, and crucially, key glucose transporters, specifically SLC2A3, which codes for GLUT3.
Hold on there. The hypoxic AT, it sounds like the proliferative AT, go, 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 and the hypoxic AT, hunker down, survive stress, are fundamentally like opposite choices for a cell, biologically speaking. Is that the kind of trade-off AAnet is designed to reveal, these opposing specializations?
That's precisely it. You've hit the nail on the head. AAnet isn't just making a list of cell types. It's mapping the specialized choices and the biological tensions within the tumor. A cell's state is defined by these trade-offs. If a cell is all in on proliferation, blue, it really can't simultaneously be optimized for hypoxic survival, orange, and vice versa. They represent different functional poles.
Fascinating. Okay, so what were the other 2?
Right. Number 4 was the immunostimulatory AT, purple. This was interesting because it involves the cancer cells themselves producing a lot of immune-related messenger RNAs, things like HLA molecules, certain cytokines. It suggests these cancer cells are actively signaling or interacting with the immune system. Often these cells seem to be paused in the G1 phase of the cell cycle.
Okay, purple is immune interaction.
And finally, number 5 was the cell damage death AT, amber. This one seemed to capture variation mainly coming from genes encoded on the mitochondrial genome. It could reflect cells that are technically damaged during the experiment, or perhaps cells genuinely undergoing stress or death pathways, which often involves mitochondria.
Okay, 5 roles. Blue growth, yellow oxidative energy, orange hypoxic survival, purple immune talk, and amber damage death. That's quite the ecosystem. So what happens when these cells metastasize, when they move to new spots in the body? Do these specialized states stay the same or do they adapt? What did they find across the different metastatic sites?
Well, generally speaking, the same basic set of ATs were found in the metastases, which is interesting in itself. They were conserved overall. But, and this is key, the proportions changed depending on the environment. Lymph node metastases, for example, looked pretty similar to the primary tumor in terms of AT mix, highly conserved. However, the liver, a really common place for breast cancer to spread, showed something different. It had a noticeable lack of that oxidative AT, yellow. Instead, it seemed to rely more on the proliferative blue and immunostimulatory purple states.
Interesting. And the lung results, you mentioned those were particularly striking.
Oh, absolutely, because
Okay.
the lung, as you know, is a very oxygen-rich environment, right?
Right.
And guess what? The hypoxic AT, orange, the one specialized for low oxygen, was completely absent in the lung metastases.
Wow.
Yeah. The environment essentially just pruned away the cell state that wasn't needed for survival there. It wasn't advantageous anymore. And even more telling, the lung metastases actually developed a unique, new oxidative AT, sort of a modified yellow state, perfectly adapted for that high oxygen environment.
So it really shows the physical environment is actively shaping which specialized states thrive.
Exactly. It's driving the selection of the cancer cell states that survive and grow in that new niche.
Okay, so moving beyond just the gene expression profiles, the researchers then used spatial transcriptomics. They wanted to see where these ATs actually live inside the tumor. Location, 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 as they say. What did those spatial maps reveal about the tumor's internal organization?
The spatial organization really confirmed the functional roles we just discussed. It was quite striking. The proliferative AT, blue, the growth engine, it formed the main bulk of the tumor, but it was specifically absent from the centralmost regions.
Okay, so not in the very middle.
Right. And the hypoxic AT, orange, the survivalist, it was found precisely in those central regions and also some peripheral spots, but it was physically separated from the proliferative and the oxidative cells. They occupied distinct territories.
Like distinct neighborhoods within the tumor.
Pretty much, yeah. And the immune stimulatory AT, purple, often seemed to form a kind of boundary layer, sort of demarcating or surrounding these different hypoxic and proliferative zones.
So the ATs aren't just abstract concepts from the computer. They correspond to real, spatially distinct functional zones within the tumor. And presumably, this specialization extends to the surrounding cells too, the microenvironment.
Yes, absolutely. AAnet was also used to identify archetypes within the tumor microenvironment, the MEATs. Things like stromal cells, myeloid cells, neutrophils, and they found really specific associations. For instance, that highly metabolic proliferative AT blue tended to be found near myeloid and stromal MEATs areas likely rich in nutrients and blood vessels. But crucially, the hypoxic cancer AT, orange, specifically co-localized with the neutrophil MEAT.
Hypoxic cancer cells hanging out with neutrophils. Why is that significant?
It suggests this idea of metabolic mirroring, or maybe symbiosis. The low-oxygen cancer cells, which are highly glycolytic, burning sugar without oxygen, seem to be partnered up with microenvironment cells, the neutrophils, that also show signs of being glycolytic. They might be creating this sort of protected, metabolically coupled niche.
Okay, that raises a huge strategic question. If you have this hypoxic niche locked into the specific way of making energy oxygen-independent glycolysis, it must have a vulnerability, an Achilles heel. The researchers zeroed in on GLUT3. Why GLUT3 specifically? Why was that identified as the potential therapeutic weak spot?
Right, so the hypoxic AT, as we said, relies on glycolysis. It uses enzymes like PDK1 to deliberately block pyruvate, a key molecule, from entering the oxygen-dependent energy cycle, the TCA cycle. So it needs to get glucose in efficiently. Now, these cells did express GLUT1, another glucose transporter, but the researchers focused on GLUP3, encoded by the SLC2A3 gene, for a couple of really critical reasons. First, GLUT3 normally isn't found in most tissues. It's mainly restricted to the brain and sperm. Finding it in breast cancer is already a red flag. Second, and this is the kicker, GLUT3 has an exceptionally high affinity for glucose. It grabs glucose much more effectively than GLUT1, maybe 3 times better.
So in that tough, nutrient-poor, oxygen-starved, hypoxic corner of the tumor, having a super high-affinity transporter is like having the best possible vacuum cleaner to suck up every last molecule of glucose fuel available.
That's a perfect analogy.
Yep.
Yes, it's the perfect specialized tool for scavenging glucose when supplies are low.
So the hypothesis was, target that high-affinity lifeline, and you might cripple these survivalist cells. Did they prove it?
They did. First, spatially, they confirmed it. Using imaging, they showed that the GLUT3 positive cancer cells were significantly closer to cells expressing HIF1A, which is the master marker for hypoxia, than they were to blood vessels, marked by CD31. So GLUT3 cells really do live in that nutrient-starved, low-oxygen niche away from the main supply lines.
Okay, so AAnet didn't just find a random marker. It pinpointed a dependency linked to a specific location and a specific metabolic state. What happened when they actually blocked GLUT3?
The functional validation was really quite conclusive. When they knocked down the SLC2A3 gene to reduce GLUT3 levels in the lab, they saw a drastic reduction in glucose uptake, as expected. Basal glycolysis dropped. Tumor sphere formation, which is a lab measure of a cell's ability to initiate new tumors, was significantly impaired. And most importantly, when they did this in vivo, in animal models, knocking down GLU3 significantly inhibited the growth of metastases, specifically in the lungs. And they even got a hint about the mechanism. Using some computational methods to infer regulation, the score suggested that the hypoxia signal itself, the presence of HF1A, actually precedes and likely regulates the switching on of the GLUT3 transporter. Low oxygen triggers the need, and the cell turns on its high-affinity glucose grabber.
That's incredibly powerful. So AAnet provides the map, identifies the specialized survivalist state, defines its specific metabolic addiction GLUT3, and then experiments prove that targeting that addiction actually limits metastasis. And you mentioned this isn't just a TNBC thing.
That's correct. A really important finding was that they can use AAnet to map analogous hypoxic and proliferative metabolic ATs across human samples of not just TNBC, but also ER-positive and HER2-positive breast cancers. It shows the tool has broader utility. It's providing a functional classification that seems to cut across those traditional hormone receptor subtypes. It classifies tumors based on what they do metabolically, not just what receptors they happen to have on their surface.
So wrapping up this deep dive on AAnet, it really feels like a massive leap forward for precision oncology, doesn't it? It takes this incredibly noisy, complex single-cell data and transforms it into something interpretable, these geometric archetypes. It shows us exactly how cancer cells specialize within that continuum and, crucially, where their hidden weak spots might lie.
I think that's exactly right. The really crucial insight here, the clinical potential, comes from moving beyond just looking at the average tumor cell, which is what bulk sequencing does. We can now pinpoint these spatially localized, functionally distinct vulnerabilities, like that GLUT3 dependence, specifically within the hypoxic AT niche. And it gives you the map and points to the location, and it suggests the target. It allows us to think about hitting the specific specialized cells that are responsible for things like survival under stress and metastasis.
Yeah, that really changes how you might think about treatment strategy before you even start. We learned that the microenvironment, like the high oxygen levels in the lung, can basically prune certain cancer cell states. So if the metastatic site itself dictates which archetypes are needed for the cancer to survive there, how might really understanding these specific cancer AT interactions with their local microenvironment change pre-treatment planning? Could we ensure we're targeting the right subset of cells in the right location for that patient's specific pattern of spread? And thinking ahead, what kinds of specialized, maybe treatment-resistant ATs might actually emerge specifically in response to therapies like chemotherapy? That's definitely something for you to mull over until our next deep dive.